The world population is currently 6.8 billion people. Scientists expect the human population to reach 8.3 billion by the year 2030 and 9.5 billion by the year 2050. To feed our growing global population, we will need to increase food production by 50% by the year 2030. At the same time, the planet's land and water resources are being stretched, so we are challenged to feed more people using less land. Scientists have used biotechnology to advance our productivity for thousands of years, as far back as 4000 BC, when Egyptians started using yeast in bread. We believe that by using the tools of biotechnology, plant breeding, and agronomic practice improvements, we can help farmers double their year 2000 yields by the year 2030. In the next few minutes, we'll hear from scientists and industry experts to help us better understand biotechnology and the value it brings to our global population. Our industry experts are Dr. Roy Fuchs, the Global Oilseeds Technology Lead at Monsanto Company, Dr. John Soper, Senior Research Director of Soybean Product Development at Pioneer Hybrid, a DuPont business, and representing government expertise is Dr. Ben Matthews, who is a research scientist for the United States Department of Agriculture, Agricultural Research Service.
Biotechnology helps us increase crop yields on a per acre basis and minimize the use of chemical agents and labor used to control insects, weeds, and disease. When costs decrease and yields increase, we make progress toward achieving our productivity goals. For example, scientists might increase yield in a soybean plant by genetically modifying it to produce more seeds per pod, by directing more energy from the sun into seed production. Our scientists might develop a trait to reduce the impact of a leaf pathogen on the plant's photosynthetic activity, which robs a plant of its yield potential. In some plants, scientists have introduced traits that naturally control pests, which reduce the need for chemicals. At Monsanto, 2 new technologies that will be introduced in 2010 will directly benefit farmers. One is solely focused on helping farmers achieve higher soybean yields. Another will combine 8 biotechnology traits in corn that will help reduce insect and weed damage and lead to increases in corn yield.
Biotechnology can enable more efficient research. For example, this Minimax is an experimental soybean variety developed by USDA scientists for research. It is smaller than a typical soybean plant, so it takes up much less space for laboratory experiments and is a good teaching tool for the classroom. Not only is it shorter, but it also matures much more quickly than commercial varieties. The shorter seed-to-seed generation time enables more genetic experiments to be conducted each year. In the classroom, experiments using Minimax can be completed easily within 1 semester. Another way biotechnology makes research more efficient is demonstrated by this fluorescent soybean root. Soybean roots are transformed with a vector containing the gene encoding green fluorescent protein. Only the roots are transformed. The stem and leaves of the soybean are not affected. This enables more research in less time because we only need transformed roots to test DNA constructs that control nematodes, which are microscopic worm-like pests. Using this method, we can have roots large enough to add nematodes for testing in less than 2 months, while it generally takes 6 to 9 months to regenerate soybean plants.
Scientists are able to enhance plants in many ways. Because these plants end up in our food supply, we value the safety of our science and the integrity of the plant. Biotechnology is a very exacting science. We identify and fully characterize the traits and their associated proteins that we're creating. We conduct extensive testing to ensure that none of the proteins are toxic to humans, livestock, nor the environment. Not only are these products safe, but we're actually creating biotech traits that can bring additional benefits to human health and to our environment. For example, at Pioneer, we're currently working on soybeans that offer a solution to trans fat. Because they are composed of approximately 75% oleic acid, they are stable oils that do not require hydrogenation, the process used with traditional soybean oil to stabilize them, but which results in the production of trans fatty acids. An additional health benefit is that the oil will provide a 20% reduction in saturated fat in comparison to commodity soybean oil. In addition to improved nutritional quality, the enhanced stability of the high oleic oil benefits the food industry by offering higher heat stability for frying and extended shelf life for manufactured products. High oleic soybean oil also provides a sustainable, environmentally friendly alternative for industrial applications, such as lubricants, foams, and adhesives.
Foods derived from biotech crops are subjected to more in-depth scrutiny than any others in history. To date, hundreds of studies have found no significant differences in biotech foods as compared to conventional foods. Many organizations of worldwide preeminence have agreed that biotechnology foods are as safe as their conventional counterparts. These organizations include the American Medical Association, the American Dietetic Association, the United Nations Food and Agricultural Organization, the World Health Organization, the National Academy of Sciences, the British Medical Association, and the French Food Agency. Foods derived from biotechnology have been eaten by billions of people without a single documented health problem. This is a remarkable food safety record, but not surprising given the pre-market scrutiny and testing of biotech crops and foods. Finally, I think we can all take confidence in knowing that the scientists and industry leaders who develop biotechnology are consumers too. Why would I develop a food product that was not safe for my family and friends?
There have been many scientific studies documenting the safety of biotech corn and soybeans for use in animal feed. For example, a variety of biotech maize received a positive safety assessment from the European Food and Safety Authority in December 2009. Also, an article in the January 28, 2009 issue of the Journal of Agricultural and Food Chemistry provides another example. This article documents that researchers were unable to detect any evidence of transgenic sequences in cattle that were fed biotech corn over a 2-year study.
Biotech crops work their way through a rigorous, complex regulatory system. A company must provide extensive data, results of field trials, and other research, as well as the company's own safety assessments, to authorities around the world, including 3 in the United States: the United States Environmental Protection Agency, the Food and Drug Administration, and the United States Department of Agriculture. Biotech products do not enter the food supply until these agencies have affirmed that they are as safe as conventional crops. Biotech seed manufacturers like Monsanto and Pioneer want their products to be developed under regulatory scrutiny to ensure safety for humans and the environment. The reality of modern agriculture dictates that this scrutiny makes not only for good science, but also for good business.
You may be wondering how biotechnology actually works. There are multiple approaches, but the most common are the development of transgenic traits and the use of molecular markers to track and select both native and transgenic genes. High oleic soybeans that I spoke of are one example of transgenics. Other examples of transgenic solutions include the herbicide tolerance and insect resistance traits that have been incorporated into numerous agricultural crops. Molecular markers allow us to see into the genetics of the plant, You could think of it as plant forensics. We can actually determine the genes that control the expression of various traits and use markers to track the genes that control these traits. Using the markers, we can match up genetic profiles of parental lines to make the best combinations, and we can ensure that selected progeny possess the required genes to deliver the traits that growers demand. Once we identify progeny that have ideal genetic combinations, we confirm the presence of those traits in our field trials and ensure that the products will meet the grower and consumer demands.
In my lab, we use laser capture microdissection. First, notice that the nematode is within the root. We use the laser capture tool to collect the cells upon which the nematode is feeding to see what genes are expressed in a resistant reaction to the nematode as compared to a susceptible reaction. You'll notice the hole where the cells are missing. We would collect many of these groups of cells, extract RNA, and then determine what genes are active within those cells.
Biotechnology is an exciting field, and the benefits are numerous for all of us. During my career, I've seen many improvements to the science, and I am confident that the science will only become more innovative in the future. Whether we are consuming healthier products or being more confident about our positive impact on the environment, we all benefit from biotechnology every day, and it will only get better.
I hope you've enjoyed learning about biotechnology. Not only will this tool enable us to feed a growing world population on less land, but it has been proven safe for animal and human consumption. Perhaps you'll even experience the benefits of biotechnology through research projects at school. Thank you for joining us today, and we hope you'll continue exploring online at getbiotechsmart.com to learn more about the benefits and advances of biotechnology.